So the first question is for Lieven. I'm going to ask a question to start off and then ask it to the audience and get some feedback. You mentioned the biomarkers issue at the end of your talk, that you've sat on 6 biomarkers for OncoCirca. There's an issue in terms of the length of time taken to validate. I'm wondering if this kind of omics approach or different approaches could yield, what I'm trying to ask is what type of partnerships do you envisage could reduce that time of validation? This.
So, in the case of OncoCirca, we have done obviously the serology, which I mentioned here, and we also have another group of activities, which is omics approaches in LCMS, in plasma and urine. And out of that came a group of biomarkers. We have submitted this for publication. It's in review, so let's see what comes out of that part. But the question now is, I mentioned that Janssen was developing flubendazole as a macrophylaricide for OncoCirca. And typically what is done in pharma, if we would talk about, for example, an oncology product, and you would find a biomarker that goes along with an oncology treatment, you would do the validation of your biomarker in the context of the clinical trial. And in the clinical trial for the compound, you will demonstrate what the intended use is going to be for that biomarker. Now, Janssen stopped the development of flubendazole because in preclinical studies we demonstrated that there was too much of toxicity with the reformulation of flubendazole. So, because of the stop of the flubendazole, we also stopped the development of the biomarkers for OncoCirca. Now, here is a panel of biomarkers that is going all the way through discovery up to validation in our biobank. What is needed to take those biomarkers into the next phase? First of all, I think there's going to be a need for a discussion on intended use. Where are those biomarkers bringing a difference? For example, will we demonstrate that individuals with adult worms under treatment with one of the compounds, maybe moxidectin or maybe one of the other compounds, that there is a change in life or death of those worms? How are we going to demonstrate that with those biomarkers? So what do you need? You need partners to come up with the proper setting where you're going to demonstrate the usefulness of your biomarker. Second is, you need access to clinical samples. And in the case of OncoCirca, if it is plasma or urine, that is maybe easy to demonstrate, but still it needs to be done in the proper clinical settings. If it's not plasma or urine, what is the biobank that you need, and are those biobanks available? In my knowledge, they are not available. So you need to develop a holistic view on what type of biobank do you need to demonstrate the clinical utility of your biomarker. And then the timelines for this. If you would develop an oncology marker, timeline is maybe for a phase 2 or a phase 3 trial, is maybe 3, 4, 5 years. That would be the timeline also to demonstrate the clinical utility of your biomarkers. If there is no compound in development, or that the compounds are already in the field, what's going to be the timeline for development? So those are the type of issues that are presented for OncoCirca. Now, thinking about the future, when I presented you the activities that we do for STH, if we think about the future, suppose that we will come up with a few new markers for STH or for schisto. But we have mebendazole already in the donation programs. There's nothing new that will change in clinical trials. How are we going to do the validation of those biomarkers? So my question to this field is, can we think about a pre-competitive environment where all those issues on biomarkers are brought forward, that we think about biobanking in a holistic way, that we think about timelines and think about funding?
So throwing that out to the audience in terms of some input into that, would there be any questions on that from the audience?
Hello. So not so much of a question, but rather a comment. I think, totally agree with Lieven that we need to have now much more of these sort of consortiums or sort of approach, much more holistic ways of this community dealing with diagnostic in the NTD space. Resources are very scarce. Validation approaches need to be concerted, and I think we're all in the same type of boat of trying to either validate some biomarkers that are there or not there, discard them, then also go into the development phase, and this is something that we have not even approached this morning, but I believe this is extremely important issue because it requires funding, it requires strategy there, and also to think in a much more, I would say, cohesive ways on how we apply those diagnostics when it comes to the clinical aspect, to the population. Again, that question of access, together, hand in hand with the donation programs or other type of programs that are going to come in terms of treatment. So there needs to be really a common strategy here. And I would like very much that sort of pre-competitive or even post-competitive, if we can put it this way, approach. We have to do it.
Thank you. Is there any comments from the panel in terms of what Bertie just said? Professor Mabey? No? Okay. A question to you, Professor Mabey. You mentioned community versus individual. You're looking at communities as opposed to individuals. I wanted to ask you how much input from the sovereign state in terms of those islands or those, you're trying to reach into communities. How are you trying to make the case for that?
Make the case for mass treatment.
Yeah, for mass treatment at community level.
Mass treatment
COVID setting, yeah.
People like taking azithromycin.
Yeah.
It's quite a good anti-malarial. It gets rid of your arthritis if you happen to have that. It's good for respiratory infections. So in general, coverage is good and people like taking it. And there was, I don't know if you saw last month, a publication in the New England Journal from my colleagues showing that mass treatment, single dose azithromycin given twice a year, reduces under 5 mortality significantly in 3 countries in Africa. So it's good stuff to take. Of course, the concern is it will lead eventually to antimicrobial resistance. But giving it once a year or even twice a year so far has not demonstrably done that.
Okay, brilliant. Thank you for that. Any questions from the floor? There's one there from Ian. Yeah. Yes, Tom.
It's not so much a question as a comment, but following on from some of the comments about the need to take a more sort of inclusive approach to this, and it's something I think I was making at one of these meetings maybe 2 or 3 years ago. If you take conventional pharmaceutical development in a pharmaceutical company, an awful lot of things are done at the same time in parallel. So you would be, that you are looking, for example, at how do you manufacture this, how do you, what's the supply chain going to be, how are you going to communicate things in parallel with your development process through your phase 2 and your phase 3 clinical trials. The problem in this situation, and it comes probably back to funding, is that the tendency is as a money-saving issue, you take it in a more sequential, stepwise fashion. But the fear then is, and I'm hearing it from panel members and question, the problem then is that because it is sequential, then everything gets piled up and it slows down the opportunity for bringing things in more quickly. So I think that one of the things we have to look at from a funding point of view is can we get a funding stream that works on brings all the partners together, as you were saying, but make sure that a lot of these elements of the development process become done in parallel rather than everything being done sequentially, because this is just going to slow everything down.
More connectivity in terms of the different silos that are working within a pharma company. From an academic perspective, I'd like to ask that question out there in terms of your view on the future of consortium building within diagnostics. Do you think, what are your views on that? Hajri, as an example, the future of consortium building in terms of pulling different sectors together to form an approach on diagnostics. How much do you think the countries are going to be involved in this in terms of the ELMIC settings? How much weight are they going to have in this as a funder?
Well, I just about to be honest with you about diagnostics, quite lots of different aspects about which diagnostic we should use first. We should sort this one before we bring it all together. And if you think about high endemic settings, we should think about which applicable diagnostic first. And when we do evaluation for these diagnostic, we should also bring all these together to make guidelines. And we do like platforms for this diagnostic, which place we should use these or should use this diagnostic for other place. And in terms of diagnostic, it's nowadays become more popular DNA detection, which is more reliable, more sensitive, more specific, and can be done in hours. And with these high advanced diagnostic, we make it more simple and applicable in the field, like what we did, RBA, recombinase polymerase amplification. We can done it in hour or in half an hour, to be honest with you, in the stable temperature. If we done that for other parasites, we might also do duplexes with using this test. If we done this test and we done different tests and see which the best, and then we can make it all together in one aspect.
David?
Yes, I think diagnostics, especially those NTDs targeted for elimination or eradication, becomes very important towards the end game. So as you're approaching the targets for elimination of trachoma, if you could just collect a dry blood spot and use it potentially for looking at antibodies to other NTDs, onco or schisto, at the same time, it would be very cost effective. And of course, it's in the interests of the pharmaceutical companies to support this because, as I indicated, otherwise they may go on donating their drug for many years when they could have stopped. So I think there's a strong incentive to develop better diagnostics and platforms that can be used for several different NTDs.
Thank you for that, David. And in terms of the platforms, Tegwen, I asked that you used omics data that's widely available. You mentioned open source. How do you think that's going to impact the future of these consortium
There's a huge wealth of omics data. There are over 100 Helminth genomes available online. Quite a lot also of proteomic and transcriptomic studies. So while I was looking for strongyloides data, I found lots of other things. So and just a collective analysis of those gives you immediately potential candidates. So that doesn't require any extra lab work. That data is already there. It's a bit like a digital biobank in many ways. So making use of that, I think, has great potential.
Thank you for that. Would there be any Lieven, on that digital biobanks point?
just on the omics data, I fully agree with you. And in our hands, we have also used quite some omics approaches for onco and for STH. In our hands, the omics approaches always fall in the discovery environment. Once you are done with the discovery and you need to go to the translation, that is where the big issue comes. So that's the transfer from a discovery into a technology platform. Like I can imagine that for your strongly believe this stool-based technology, you might think at the end of the day on an ELISA, right? That is where the transfer comes, and that is where the big issue is. It's not in the use of omics itself, because omics is a discovery.
Thank you for that. Any input, I think, from Poppy at the back? Yeah.
Thank you. Just one point following on from that, actually. As well, it can be used in the discovery, but also if you're thinking about cross-reactivity, then the omics database is also fantastic for searching for potential cross-reactivity. I also had a couple of just really small questions, not about STH actually, but about onco and schisto. Which species of schisto were you using, and was it species specific? And then in the onco, did you find anything that might be stage specific, so adult worms versus microfilaria?
So for the peptide microarrays for schisto, that was Mansona. But I don't know if any. I think there was an issue on space on the array. We are focusing mainly on STH, and in STH we had both Ascaris Zoom and Ascaris lumbricoides. So we decided to add more on Ascaris than on Schisto. For the topic on Oncocerca, which stage that we are using, so that brings me back to the discussion on intended use. For us, for Janssen, we approached this from flubendazole, and flubendazole was meant to be a macrofilaricide. So we were looking for biomarkers that would give an indication on how flubendazole is changing the activity of the macrofilaria during treatment. And so this was a very complex issue in a way, because you don't know how many worms, adult worms, are present, and you don't know if your compound is reaching all compartments where those worms are, and you don't know what is secreted or excreted from those worms. So in biomarkers it was a big challenge, but the main goal was to look for biomarkers for microfilaria. Now, having said that, I don't know what we discovered. Right? We have, for example, a urine marker, and I don't know what that urine marker is going to be. Is it a marker for microfilaria or for macrofilaria? I can't tell you now.